Welcome to Ask the Expert. This is a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. And we're recording this event. We're going to post it on the Sugar Science site YouTube channel shortly after the presentation. It's also available on Spotify and to listen as a podcast. And if you have any questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. And today we have as our guest, Dr. Maike Sander, MD at UCSD. She is an expert in pancreatic stem cell biology with over 20 years in this field of research. She earned her MD at the University of Heidelberg in Germany, followed by research at UCSF. And she's currently the director at the UCSD Pediatric Diabetes Research Center. She's an elected member of the American Society of Clinical Investigation and a member of the NIH Beta Cell Biology Consortium and a recipient of the prestigious Grodsky Award from JDRF. So she's quite busy and quite at the top of the field. She's widely published. She's cited with 165 research works with 8,958 citations. People are listening to what she does, and her new review is deeply informative of the intersection of genetics and T1D. The review is titled Transcriptional Mechanisms of Pancreatic B Cell Maturation and Functional Adaptation, and it came out in Trends in Endocrinology Metabolism in July 21, so this month. So welcome, Maike, and I am really excited to talk with you today and hear what's going on in your lab down at UC San Diego. A lot of really interesting genetics work is coming out of that university. And so I cannot wait to hear what's going on.
Thanks, Monica, for the invitation. This is a great forum. So what I'm going to do in the next 10 to 15 minutes is comment a little bit on what we actually do in the lab, and then give you a few vignettes of what we are really most excited about. And I would say what really excites me most about research now, and also the place that we're at in type 1 diabetes research, is that the research has become highly interdisciplinary. So we really have embraced that as a lab. So my background is in beta cell biology, and I've spent sort of the 1990s and 2000s to dissect much of the gene regulatory circuits that underlie the formation of pancreatic beta cells. And then really having studied that in much depth, that provided the blueprint for instructing stem cells to make beta cells. So that transition really going from mouse genetic modeling to human iPSCs was sort of a natural one, with the basis that we and the entire field had laid really in the 90s, 2000s, and still ongoing. So in the lab, we combine a genetic mouse model still, because it is really the hallmark physiological model in the end, because you can only really model diabetes in an animal. You can't do it in a dish. You have the readout still, does that organism actually manifest diabetes? So I think animal modeling still has a huge place in research there. Yeah. But then, of course, we want human models, so we combine that with this human iPSC model, and I'm going to talk about this a little bit more, in that we have really learned how to make beta cells in the dish. And then a huge part of the lab is engaged in the sort of right arm of the slide here, where we combine genetics and genomics with this human iPSC model to really understand the underlying gene regulatory defects that lead to beta cell dysfunction, and then also beta cell death in type 1 and also type 2 diabetes. So on the right side there, I've just highlighted some of the work that we published this year collaboratively with Dr. Kyle Galton's lab, who's also here at UC San Diego. So Dr. Galton, I think, has also spoken in this forum, and he has really pioneered many of the much of this research in genetics and genomics, yeah, as it relates to the pancreatic beta cell. So we teamed up with the Galton lab to then use our human iPSC modeling ability to really validate the hypotheses that arise from generating these genetic maps. So ultimately, you want to also see what's the functional relevance of the genetic hotspots that you identify in the genome. And there's a lot of ongoing work where we do this collaboratively. We're also working collaboratively with immunologists to really begin intersecting the beta cells with the immune counterpart and using this human iPSC model system to begin to model the autoimmune attack on the beta cell in vitro. And much of that work we're doing here with Luc Titan, who is a type 1 immunologist at Scripps Research in San Diego. So, as said, much of our research is multidisciplinary. You really can't do things alone anymore. And that is also a huge opportunity. And for me as a scientist, this is really probably the most exciting thing about the era we are in, because you're constantly learning from others. You're learning about new technologies that other fields develop. And then we as diabetes experts know the questions to then really think about how you can innovatively apply these technologies to find answers to these questions that we've had for decades, ever since we've known about the disease, is the exciting part. So what I want to focus on today in a few vignettes that I want to share is really our research into building better models to model diabetes using human pluripotent stem cells. So I'm sure others have spoken about this in this forum. So the entire field, really building on decades of mouse genetic research, has allowed us to then develop recipes in the dish so that you can instruct human pluripotent stem cells to make beta cells and pancreatic islets. So on the right here, I'm showing sort of a stem cell derived islet that we are making in the dish. And it really has all the major endocrine cell types. And from that, it actually looks remarkably similar to a primary human islet that I'm showing on the bottom here. But functionally, we still have deficits. So we know we can transplant these islets or stem cell islets into mice, and that they do mature in vivo into functional beta cells and can cure diabetes. And you probably all know that there's clinical trials ongoing with stem cell derived islets, and that's a very active, exciting field for type 1 diabetes research. What we are focusing more is on the modeling aspect, really using these stem cell islets to begin to identify mechanisms that lead to diabetes and beta cell demise. And Monica has mentioned this review that we published just recently in Trends in Endocrinology and Metabolism. And there we really discussed what we understand about human beta cell maturation and the environmental cues that are necessary to mature a beta cell in the first place. And there we raised sort of conceptual ideas as to how we might apply these principles to then really mature these beta cells also in vitro, because there is still a missing piece. Yeah. So then when we think about these stem cell islets, how good are they? What can we model? What can we not model? And that's what I want to share in the next 5 to 7 minutes with you. So the stem cell islets that we make, they have all the major cell types. But when you look at a primary human islet, it's not just endocrine cells. You have endothelial cells, you have blood vessels going through, and we all know the islet is highly vascularized because the hormones are released into the capillaries and the bloodstream. So each endocrine cell really abuts a blood vessel and a capillary. But we also know that there is cell-cell signaling that occurs between blood vessels and endocrine cells that is important for the function of these endocrine cells. So an important piece that we are lacking in the dish is really these blood vessels. And we sort of came up with this idea, could we maybe build a stem cell derived islet where this stem cell organoid is not nourished by just sort of floating in a tissue culture dish, but maybe where we could build vessels into this organoid so that these stem cell derived organoids would actually be nourished by blood vessels through which we could supply the nutrients. So having blood vessels in play is not just important for the cell-cell signaling and the overall physiology of the beta cell, it's also an integral part of understanding type 1 diabetes, because we know that in type 1 diabetes, the islet becomes infiltrated with immune cells and then T cells that ultimately destroy the pancreatic islet. And that a normal islet is actually engulfed in like something that we call an extracellular matrix. So it sort of has a capsule, and all of this we're really not recapitulating in the culture dish. And all these components are important for the immune infiltration, because we know that the immune cells sort of penetrate this extracellular matrix, that blood vessels are an integral part, and that actually in humans, blood vessels themselves can serve as antigen presenting cells, so could well participate actually in initiating the autoimmune response. So it all really started with a collaboration that we had with Chris Hughes Lab from Korea University, where there was a graduate student there, Yesel Jun, who then joined my lab as a postdoc after she graduated, where what Yesel observed in her graduate work was when you put these islets, and she did this in rat islets, so I should say that in a normal tissue culture model, even of primary human islets, these endothelial cells and the blood vessels, and also the immune cells, they quickly die. So whenever we have like a human islet model, we're actually modeling things without this important component of the vessels of immune cells, because they don't survive in these tissue culture conditions that we developed for islet culture. So what Yesel did in this work is that she put these islets into these microfluidic devices. So they're like miniature tissue culture chambers, where you can now not just have a static culture, but you actually apply flow to the tissue culture. So that's really what cells are experiencing in the human body. So it's really more in vivo mimicking conditions. And under these conditions, what Yesel saw is that the endothelial cells, so the blood vessel component, that these cells survived, and that also the beta cells functioned better. So that gave us clues that really the flow is an important component for beta cell function, and as well as endothelial cell survival. So we then undertook this really endeavor, because we've been doing this for 6 years now, to figure out how to vascularize these iPSC islets that we can generate in the culture dish. So in order to monitor beta cell health, ultimately in something like this microfluidic device, we needed a real-time readout for beta cell function. And we generated this by using a calcium reporter. So calcium influx leads to insulin secretion. So these calcium reporters can be used as a surrogate for insulin secretion. And we introduced that in human iPSC. So that is a real strength of this ESC iPSC system, that you can genetically manipulate it and not just test the function of genes, but also introduce these beautiful reporters. So what we observed then is that this reporter, these iPSC or ESC beta cells, responded nicely to actually, they responded to exendin 4 with calcium influx, with potassium, so that those are normal triggers of insulin secretion and calcium influx. But really the response to high glucose is still relatively flat. So this is still like a deficit that we observe in these ESC, iPSC beta cells, that not just our lab has observed, but also others. So there's a landmark paper right now in bioarchives from Timo Otonkovsky's lab, and they describe that same sort of lack of calcium response. So there's clearly things we're not doing right in the dish yet. So then we thought the endothelial component of the blood vessels could be one of the important missing components. So Yesel and we in my lab really set out on a painstaking kind of journey to identify or culture conditions that really allow these iPSC, ESC beta cells and islets to be vascularized. And for those of you working in tissue culture, every cell type has its sort of culture medium and culture composition. So they took a while to identify culture conditions where all these cell types happily coexist and could actually be cultured together. So we can now generate these iPSC organoids. You have in green here the insulin positive beta cells, and then in red, these CD31 positive endothelial cells that are budding the beta cells. So the first question we asked in this model was really to see, do beta cells function better when blood vessels are present? And this was indeed the case, that now we can, in response to high glucose, really see these calcium excursions and spikes. But we only really see it in the cells that abut these endothelial cells. So this is really validating now in a human organoid system what people have observed in mice, that blood vessels are important for beta cell function. So we've teamed up with Chris Hughes Lab, and they developed this beautiful microfluidic system where they can actually create living blood vessels.
We actually just had Hugh Bender the other day, and he was talking about it. It's fascinating. I love that you're collaborating with them.
So yeah, so we have worked together. It's been a really wonderful collaboration now going on to 6 years, where you know, they have really pioneered just like creating real blood vessels in the microfluidic device, where you have an artery, and then you really feed the culture tissue, the tissue culture medium through these vessels. So they're actually non-leaky vessels through your tissue, and then you have a venous return, just like in the human body. So we thought if we could actually, Hugh and Chris, so they have really pioneered this now for seeding primary human islets into this, and they're doing studies with that. But we wanted to then take it a step further and put these stem cell derived islets that we already learned how to vascularize in vitro into this device, and ask whether now adding this flow through the device and these vessels could further improve the functionality of the beta cells. And that's indeed what we found. So now you really see these nice kind of calcium spikes in response to high glucose. So we now pretty confident that we actually have an organoid that's vascularized that we can put into a microfluidic device and begin to model type 1 diabetes in an actual vascularized context. So
that's really remarkable. That's a remarkable difference from the earlier slide.
Yes. So in the last slide, I just want to outline where we're going with that. So now we're actually bringing our immune collaborators into the fold, with whom we've already been working, because we know that the immune component is very important to the pathogenesis of type 1 diabetes. So we have generated iPSC lines from well-phenotyped people with type 1 diabetes. And we have actually these people on recall for blood draws here in San Diego. So we can always harvest fresh immune cells from these individuals. So we now can make the endothelial cells, the beta cells, and the stromal cells, all with a genetic makeup of these people with type 1 diabetes, create these organoids, and then begin modeling the immune attack on beta cells in this vascularized context.
That is such an amazing project. And I mean, that's so much fun for someone to get in there as a postdoc or a graduate student and start digging into that system.
Yes. So it's been a terrific collaboration, and I'm just outlining here the many who have contributed to this work. I think I mentioned the people on the slide, so I'm not going to go through everyone, but I just want to really leave everyone with saying, you know, science is a team sport these days, and this has also been generously supported by NIDDK. So we are part of this HERN consortium that actually fosters multidisciplinary research, and that has really funded our effort to develop these organoids on a chip. So I'm going to stop sharing here and happy to start the discussion.
That is just some fantastic work. Really, really interesting. And, you know, it just has the whole idea that you're bringing in the sort of the real clinical situation from people who really have type 1 diabetes and are now able to test it in this setting is, I think it's really going to blow the lid off things. I just wondered if anyone here would like to ask a question. I see a few big names in the audience, so that would be fun. Anybody?
Sure, I'll ask a couple questions.
Okay.
Sure.
Hi.
Hi. So this is amazing work. This is really great. Exactly kind of what we needed. And I'm glad that you looked at the blood vessels before you put a capsule on, because without the blood vessels to perfuse what's inside a capsule, I'm not sure that there's going to be a lot of oxygen in there. So that's probably the right order to do things. I was thinking that maybe those endothelial cells are also presenting antigen. You mentioned that they present antigen in the human, and I thought maybe they were presenting antigen in a non-inflammatory environment to promote tolerance, because it's certainly true that the body maintains immune tolerance by presenting antigens in a non-threatening environment. Cross-presentation. And so I thought that was an intriguing possibility as well. So thinking of, we always think of two sides of the diabetes coin, one being the side where the immune system is attacking, the autoimmune attack, and the other is the active tolerance. And so this affects that as well. With respect to the peripheral immune cells that you're getting from these type 1 diabetes donors, our problem has always been we've had two problems in type 1 diabetes research. One being, of course, that the pancreas is not very accessible in the human at all for study. NPODs address this partially, but still it's a difficult problem. But the other is that the T cells that are lymphocytes that are doing a lot of the work of attacking the beta cells are not present very much in the peripheral blood. So they go to they go out of the thymus, they circulate a little while, and then they go to the target organ or its draining nodes, and they're just not accessible. So I was wondering, my question to you is sort of how you might address that problem. I know that people have isolated islets in, for example, NPOD, and then eluted the T cells from them. So that might be a more interesting sort of, it's very difficult technically to do, but it's been done now repeatedly. And so that might be a way to approach it, but you might have other ideas.
Yeah, no, so this is really, I think, when you think about now, using this model to gain insights into type 1 diabetes, whether it's the effector phase or, even harder to study is what actually initiates it. I think we can of course use pre-existing T cell clones and use those, but you might actually not learn much about, what kind of T cells are actually present, and we don't really know, now that we are actually thinking about transplanting iPSC beta cells into people. I think a really important thing is to also start to think about that memory compartment and what might actually, even an autologous graft, right? What might attack that autologous graft. And as you say, it's the needle in the haystack. These immune cells are not abundant. But one thing that we are working on with the Teton lab, so, I'm not the immunologist, is to really use these iPSC beta cells now as a bait to maybe re-amplify those rare cells so that, your needle in the haystack kind of gets hopefully amplified, and we might be able to gain some insights into that memory T cell compartment by actually having the stimulus, the antigen, because it might just be that because in a lot of people with type 1, there aren't like many beta cells left. So for that reason, there might not, the T cells might be so rare just because the stimulus is missing.
Have you guys, this might be a really, kind of naive question, but what if you sort of like created that bait, like a patch, with GAD or some kind of autoantibody to the skin of these patients, and then, maybe lured some of the T cells out of their sort of senescent or quiescent state in some of the nodes where they're just sort of like sleeping quietly, lured them to the site on the skin, and then isolated them from there and grew them up. Is it possible, or is that just totally out of the realm?
That I don't know whether that would, it's like you need the antigenic stimulus, right? Like, the
GAD65 and some, IA. I don't know. This is just throwing it out there.
Yeah, we don't even know which T cells. Ultimately, it's the T cells probably that are the destructive part. Would anti-GAD ones be destructive? There's a lot of work on that in the NOD mouse where people have actually looked. Right? Is you see the autoantibodies. We know we have reactive immune cells against that in people with type 1, but which ones are the ones that actually kill the beta cell?
Yeah, because some people are saying GAD65 is protective, but yeah, like if maybe you could do like just a couple of little patches and see what see if you could lure them out of the nodes. I mean, that's really far-fetched, but maybe you could isolate some of the bad guys that way. I don't know.
So Bill, what do you think about that?
I think that might be a little too artifactual. I mean, so what we
Yeah.
really don't want is any kind of an artificial or amplified cell that would attack GAD in an islet, because a great model system, but may not have much relevance to reality. So, I mean, it's a great idea, but I think it's probably not the right approach to go. I was captivated with what you said, Maike, about the viruses, because, you know, I think a lot of people study, this is going to be my hand waving, if that's okay. A lot of people study the reaction of the immune system against the beta cell antigens. And, you know, this has been a focus of a lot of people's thinking. But if you think about what might be triggering and driving diabetes, which has a long, long prodrome, it might be the antiviral response, for example, that might be driving that. And there's a couple of things about that you should think about. Number one, if you infect your system with viruses, you can kind of see for which viruses the welcome mat of the beta cell is actually out. I noticed when you showed your green fluorescent protein that you got blocks of beta cells that were depolarizing at the same time, right? And why is that? It's because there's a gap junction protein that actually
there's beautiful work on that looks at the connections and all that. Yes.
Yeah, and you know what the gap junction protein is? It's not
the
named
entryway for the virus.
It's not named as a gap junction protein. It's named as a Coxsackie adenovirus receptor, the CAR protein, right?
Yeah.
And so there are a set of viruses that use that as a welcome mat to get into the beta cell, and this would be a perfect in vitro system to be able to look at that. And then you also could look at, perhaps, again, you know, I know that there might be artifacts here, so it may not be completely relevant, but you know, whether or not these viruses can set up shop chronically in the beta cell, which is a key. You know, prolonged infection is a key idea. So, I mean, this is a perfect system to kind of study that. And then the final thing I would say is that, you know, whereas the peripheral immune cells that are reactive to GAD or IA2 or zinc transporter or whatever are probably in very low precursor frequency in the peripheral blood, it may be true that the cells that react against Coxsackie virus or adenovirus or whatever are maybe more plentiful in the peripheral blood. So you could probably get those, and I get those from a diabetic because they had the diabetogenic virus, presumably, earlier.
Yeah, I mean, what about the COVID people, the patients who went on to present with type 1? Would that be a good set of blood donors?
I don't know that literature well, and I'm not a big fan of that.
No.
I actually, you know, I don't. The level of what is it, ACE2 on the surface of beta cells, it's there, but it's not there in high amounts. I think the elephant in the room is CAR and Coxsackie and adenovirus. So sorry.
No, I just wondered because this morning there was a big, you know, Camilo in University of Florida. Camilo, I forget his last name. Oh
Ricordi.
Thank you.
Ricordi, Miami.
Yes, in Miami. Sorry, he posted this whole thing, like 40% of people with diabetes that, you know, have now 40% of people with COVID now have come down with diabetes. So it was a big post, and I was kind of like, you know, I think it's mostly like type 2, but, you know, maybe the type 1s could provide some information.
I think that's still like a huge gray area, and there's a lot of rushed research we've seen over the last year. So
yeah, rush for funding. So but yeah, no, anyway, I'm just throwing these things out there, and thank you so much, you know, Dr. Hagopian, for weighing in and, you know, kind of adding your long lens of understanding in this field to sort of some of my naive questions. So I appreciate that so much. Yeah, so I wonder if anyone else would like to ask a question. Keith, I know you're in organoids. Anyone else?
Thanks. I didn't really have a question. I just would say I've done a lot of work in the organoids, which I see you're clearly getting into in the right way, and it's impressive. So I would say that it's definitely like my work has been. I'm a CEO of a couple of biotech companies based in San Diego, focused in bioprinting, and we've built things like NASH models using diseased patient cells and incorporating endothelial cells. What we haven't done is go to this next step to have the blood vessels better represented, and it's definitely the right place to go, especially in the context that you're discussing where there's some active role of the vasculature. We've tried to take advantage of areas where the sort of passive diffusion, added passive diffusion, and the signaling role that the endothelial cells play, adds a lot of value. And so we've seen that. But if you're thinking, when we think about going into other areas, this is a great example. Or if you're trying to model certain aspects of oncology, and having a perfused tumor or perfused vasculature is is very attractive. And so I think this is exactly the right way to go, and it's impressive. It's a great next step.
Thanks. Yeah, bioprinting is one thing we've also discussed that we haven't really applied to it, but that could actually also add value to it.
Yeah, and I should
have another conversation offline.
Yeah, and I can comment. I mean, it's kind of the right tool for the right job. And what we realized when we built our first 3D liver model is that we'd probably skipped 3 steps, and that spheroids, could have gotten us some things if we had started with that. And now there's more people in the space building, the cell aggregate spheroids and getting, I think, a level of value where the bioprinting will come in more importance is when you need specific structure and getting cells into the right, interactions in space with the right signaling back and forth and the right mechanical signaling. Because when you have a spheroid, typically then you're not attached to the plastic. And so the mechanical signaling is also better. I think that all adds up to a lot of value as a first step. And so you're going to get a lot of value. And then if you think you need structure, like in the liver, we needed an interface between hepatocyte and stellate that had to be created. You can't just randomly mix them. And so that's why we needed structure.
Thanks.
It's good to know you guys are all down building incredible things in UC San Diego. I think it's really poised down there to make some big splashes with a lot of the biotech that's coming out of there, and companies that are coming out of there. Arcari is one of them, and others. So thank you again, Maike Sander, Dr. Sander. I really appreciate you speaking with us and entertaining these questions. And we just can't wait to see what comes next with your new systems.
Thank you.
Okay, have a great rest of your day.